Hello, everyone, and welcome to today's DDN webinar. I'm Sunita, Assistant Science Editor for DDN, and I'll be moderating a discussion. We have an exciting webinar planned for you. Our speaker, Dean A. Lee, will discuss the unique advantages of using natural killer cells for chimeric antigen receptor therapy and the latest developments in their clinical utility. After the talk, Dr. Lee will participate in a live question and answer session. Please submit your questions or comments to the Q&A portal to the right of your screen. We will try to get to as many of the questions as we can. I'd like to take this opportunity to thank our webinar sponsor, BPS Biosciences. BPS Biosciences' purpose is to advance new scientific discoveries that lead to therapies by creating innovative solutions for research. Their focus areas are immunotherapy, genetics, coronavirus, CRISPR, cell signaling, and others, providing 4,500 plus high-quality recombinant cell lines, lentiviruses, proteins, and biochemical and cell-based assay kits. As a primary manufacturer, they supply products in large-scale bulk quantities as well as contract and custom manufacturing. Their biochemical and cell-based screening services are also available to accelerate research. Our sponsor has provided us with some helpful handouts related to today's webinar. You can find and access these in our handout section located on the right side of your screen, where you can also find a certificate of attendance for participating in today's live event. And with that, let me introduce a speaker. Dean A. Lee is a professor of pediatrics at Nationwide Children's Hospital, where he holds the DeMarco Family Endowed Chair in Cell-Based Therapy and serves as the director of the Cellular Therapy and Cancer Immunotherapy Program. He is a bone marrow transplant physician and conducts clinical and translational research on natural killer cells for cancer immunotherapy, focusing on developing genetic and non-genetic methods to enhance natural killer cell function against cancer. He developed a novel approach to generating highly potent natural killer cells that have been infused into hundreds of adult and pediatric patients with leukemia, brain tumors, and solid tumors. Lee holds dozens of patents and commercial licenses. Let's just take a minute to make sure the slides are working fine. Yes, they are. Dr. Lee, the stage is yours.
Great. Thank you very much. And thanks to the organizers and Drug Discovery News for inviting me. Many of us get Drug Discovery News in our mailbox, and it serves as a really nice sort of first look for what's going on in the industry. So I thought for this talk, I would cover a little bit of the background around why NK cells in the first place, some of the history around getting NK cells into the clinic, some of the hurdles around getting genetically modified NK cells, and then sort of where we are as current state. And a few references at the end just for further reading for anybody who wants to go on a little bit further in this topic. So bone marrow transplant is really kind of the original cell therapy. What we thought we were doing was simply replacing hematopoietic stem cells so that we could give more chemotherapy or radiation. Right, the initial advances in treating cancer around drug identification really didn't take hold until we also knew how to combine and dose escalate those drugs. And the bone marrow was the limiting toxicity. You killed fast-growing cells with chemotherapy and radiation, and normal cells that are fast-growing include your hair and your bone marrow and the lining of your mouth. And so the limiting of those, everybody would easily trade being bald for being cancer-free. Well, maybe most people. I'm not bald, so I can't say that entirely. But the limiting factor is really the toxicity to bone marrow. And so if you could just replace bone marrow, then you could go higher on your doses. However, what we learned was that we were replacing an entirely new immune system with that transplant, and that we were delivering not just stem cells, but a whole host of T cells, CD4 and CD8, NK T cells, NK cells, B cells, dendritic cells, that all led to graft versus leukemia, and now in the solid tumor setting, understanding that as a graft versus tumor effect. So it wasn't just more chemo and more radiation. It was that there's an immune response to the transplant against the cancer. So NK cells are really have a core role in the center of the immune response. They are first responders to many viral infections. And as that, they interact with B cells, dendritic cells, and T cells, NK T cells. They form a bridge between adaptive and innate immune responses by setting up this micro-inflammatory response that recruits the rest of the adaptive immune system. So what we've seen often as we're learning more about what the role of the NK cells are, is that in the transplant setting, their recovery is really necessary to set up then the immune response to cancer. But in many of our cancer treatments, they're one of the most potently affected cells. So understanding this transplant as immunotherapy is really kind of obvious, even when we look back at some of the simple parts of our therapy, in that we have a graft versus leukemia effect that is traded off by graft versus host disease. So graft versus host disease is the unwanted part of an immune response, where we're recognizing normal host tissue rather than the leukemia or the cancer. And as you can see here, there's sort of an optimal amount of understanding an immune activation that leads to overall survival. So in disease-free survival starts to drop off when you have very high graft versus host disease because it's attacking your normal tissue, but then you have very little relapse. And so you've completely controlled your disease, but now your immune response has gone overboard and you're attacking self. So NK cells have kind of been seen as overcoming this trade-off because they don't cause graft versus host disease, to the best of our knowledge. They don't recognize host tissue in the same way that T cells do. They're not looking at MHC and antigen presentation as an activating signal. They more often look at MHC as a regulatory signal for discriminating self versus non-self in the opposite direction for control. So the problem is that although these have a potent anti-tumor activity and really important in promoting adaptive responses, leukemia itself, by its very fact of infiltrating the bone marrow, reduces NK cell numbers. NK cells need to be produced at a fairly consistent rate. They are generally not long-lived. Most NK cells last 2 to 4 weeks in the blood, and so they have to be continually produced by the marrow. And if that bone marrow is overwhelmed by cancer or is being destroyed by the chemotherapy and radiation, you can't make those NK cells, and so you have fewer numbers. Then there's also a whole pattern of the tumor and leukemia itself suppressing NK cells, production of TGF Beta, IDO as an enzyme that cleaves tryptophan and results in immunosuppressive byproducts such as chimeric acid, and NK cell specific checkpoints such as TIM3 and TIGIT. And then the last thing is we finally have these patients come to us, and as I mentioned, we give them chemotherapy and radiation, and we wipe out every last NK cell that's left. So this has created therapeutic opportunity where we now have many clinical scenarios where we can now add back NK cells into this NK cell penic environment. So the role of NK cells in transplant has been shown in a lot of different ways, both related to number, graft content, function, and genotypes. And so some of the earliest data was just how fast do the NK cells recover. And in this group, they looked at recovery at day 30 after transplant, and they call that the NK30 count, so how many NK cells at day 30. And fairly arbitrarily split it between those that were above and below the median of 150 per microliter. And you can see if you had more NK cells, your NK30 was more than 150 per microliter, you had less GVA, higher, you had lower mortality, you had and this survival was really most obvious in myeloid leukemias. It seemed to be less prominent in lymphoid leukemias. So initial approaches at trying to adoptively transfer NK cells were really based on collecting NK cells by apheresis. So basically spin around the blood, separate it into red cell, platelet, plasma, and white blood cell components, collect the white blood cell components in a bag up here, give everything back to the donor, and then take that product and deplete the T cells, because we don't want the GVH that T cells cause, activate what's left with IL-2, and that's your NK cell product. So the problem with that is that relatively small doses were able to be achieved that way. And there was hints at efficacy, but really difficult to do this on a large scale. So many people turned towards development of feeder cells that could do ex vivo expansion. We actually stumbled on our feeder cell, really because I was studying T cells, and every once in a while we would get overgrowth of these NK cells in our T cell cultures. And what we ended up settling on was K562 as a base feeder cell. This is a myeloid leukemia cell line that was derived in the 1970s. It's been transduced with 4-1BB ligand for co-stimulation and a membrane-bound version of IL-21. And we came across this really as being the most consistent, reliable, and robust approach for growing these NK cells. One of the key findings at the beginning was that IL-21 really helped maintain expansion in a log phase for many weeks on end compared to IL-15. We started to see that there was this IL-15 mediated senescence after about 4 to 5 weeks. It actually enhanced cytokine production compared to baseline NK cells. And this survival was mediated in part, at least, because of activation of telomerase and maintenance of telomeres in the NK cells. So with this approach, we easily get 2,000-fold expansion in 2 weeks, 30,000 plus in 3 weeks. And that means we can deliver doses now looking at up to 1 billion per kilo are really feasible, where with the apheresis approach, the average was around 10 to 20 million per kilo. So we're log orders above in terms of being able to dose escalate this cell therapy. This approach, as I mentioned, was also quite robust, not just in ability to grow peripheral blood NK cells, but in growing NK cells from cord blood, from patients, CSC, embryonic stem cells, and even across species in humans, monkeys, and dogs. It does not work very well for mouse, and so we've been separately working on trying to find a related approach for growing mouse NK cells. And so most of our studies have been limited to work with humans and some large animal models in the dog and the monkeys. I'll get to it a little bit later. So just because you can grow large numbers doesn't really matter if they don't work, but these NK cells after expansion also maintain very high function. They have very high levels of granzyme and perforin and are able to elicit cytotoxic events, as you can see here. This is a brain tumor cell line from a patient with ATRT. This is a brain tumor common in young children that's very difficult to treat. This process of recognition happens quite quickly. It's really a matter of a couple of minutes. And once the NK cell is determined that this is a target, it releases cytokines and perforin and granzyme for a targeted cell death in a matter of minutes. So our next hurdle was to get this adapted to something that could be used on a clinical scale. And what we ended up with was a pretty consistent process we've used now across almost a dozen clinical trials, where the donor blood is collected, often in a blood bank-like setting. We don't really care about much except the white blood cells. And in some cases, we even let the red cells, platelets, and plasma go back to the blood bank. But the Buffy coat fraction then is sent to the GMP, where we do CD3 depletion to get rid of the T cells, once a week stimulation with the feeder cells, and either infuse that as a fresh product or cryopreserve so that we have product on hand and can thaw and infuse multiple doses into the patient. Now, this part of cryopreservation has been really important for us to overcome as a hurdle. For many years, it was thought that NK cells had to be infused fresh. They really are sensitive to cryopreservation. We have a long history of trying to understand this, and in many cases, you would lose 50 to 70% of your viability by one freeze-thaw cycle. Fortunately, once they're expanded in this approach, these cells actually tolerate cryopreservation quite well, and we very routinely get more than 90% viability after thaw. So we've now moved almost entirely away from fresh products. The fresh products are rather difficult to arrange your GMP facility in order to time a fresh product at exactly the time that the patient needs it. And so all of our trials in the last 3 to 4 years have been exclusively on thawing and infusing cryopreserved products. So one of the first of these for us to do was to go back to sort of first principles, where we understood transplant as being a place where NK cells really mattered. And working with Stefan Churia, he had developed a platform at MD Anderson for haplotransplant that really leveraged this idea of post-transplant cyclophosphamide Several other groups had really pioneered this, including groups at Johns Hopkins. But Stefan-Turia's approach was to use a relatively reduced intensity conditioning with melphalan and fludarabine, and then give the stem cell product, the post-transplant cyclophosphamide at day 3 or 4. And what we understood about this post-transplant cyclophosphamide is that the very few numbers of NK cells that you delivered with the transplant were now going to be killed by this chemo. And so you had even worse NK cell function in this particular setting, which in part actually helped us understand that it was a better place for us to give back the NK cells. So we really layered over the top of this our NK cell part of the regimen, and we compare them against historical controls. And many other institutions had started to do this kind of post-transplant transplant approach, so that we had patients that we could compare with through the International Transplant Registry, CIBMTR. Very nicely, we were able to show in a phase 1 and 2 combined trial that in a case match control with the CIBMTR, that we reduced relapse rates. We reduced relapse from 38% to 4%, and increased overall survival from 44% to 66%. So a 50% improvement in our overall survival. And all we did different was add back the NK cells. So this has really provided for us a background understanding of how well just restoring NK cell function can be. Although we now recognize that there's not only in leukemia, but now to apply this to many solid tumors, we'll need to introduce additional targeting so that the NK cells can recognize those cancers. So another kind of important principle is that NK cells, because as I mentioned, they're relatively short-lived, 2 to 3 weeks, we do know of longer-term memory-like NK cells that survive longer periods. But it's important to start thinking about these as a drug and to think about multiple infusions of NK cells for repeated dosing, rather than just holding to the traditional CAR T cell approach, which looks at a single infusion being a lifelong therapy. That's great where it works, but it actually creates several problems. One is that you now have constant targeting of whatever your CAR T cell is going to target, which may not be tolerable for some tumor antigens. It works great for CD19, but we may want to think about this more as a drug that you give with an intended peak and trough and pharmacokinetics of how this drug is delivered. And now that particular principle we've applied also in the setting of leukemia after chemotherapy, with this idea that the chemotherapy itself, this is particularly for for AML, for refractory AML, FLAG chemotherapy is a 5-day course of fludarabine and cytarabine, and this also has a profound effect on NK cells. So could you restore NK cell function afterwards by multiple doses and sustain an NK cell response during the 2 to 3 week period after this chemotherapy is delivered? Some interesting results from this. So we have one paper that was published last year by Lucia Sila and one that's in progress. 2 different trials that have done the same approach, one in Brazil and one here in the US. One of the first patients that we treated was a very refractory patient. And without going in detail through this, you can see that he had multiple lines of therapy in order to get control of his disease, including standard 3 and 7 induction, had itosarin, mitoxantrone etoposide, clofarabine, decitabine, finally went to transplant, and then even after transplant had a CNS relapse, had cranial radiation, and donor lymphocyte infusion. So lots of different therapies, and still now he had a resistant disease. So when he came to us, on the trial, he had about 93% of his bone marrow was leukemia. We gave him the 5 days of FLAG and 2 weeks of NK cells. And our first measured time point was 1 month after the NK cells were infused, and he was now down to 5% leukemia in his bone marrow. And we thought, well, that's great. now you should go get some additional therapy, go get another transplant, go get something else for this. But his primary physicians weren't really sure he could tolerate much else, and he needed some time really just to recover from all the therapy he had. So they decided just to watch him. And surprisingly, it's not normally, we don't watch patients who have residual leukemia, but a month later, he was down to 0.5%. And then 2 months after that, he was down to 0.01%. And what this really helped us understand in this case was either the NK cells were actually surviving for quite a long time and having a continued anti-leukemic effect in this patient, or perhaps they were finally doing what NK cells are supposed to do and talking to the rest of the adaptive immune system, had set up that inflammatory microenvironment and talked to the T cells and B cells that were necessary to initiate an adaptive immune response. And it was now the adaptive immune response that was taking over. In this particular case, we'll never know which one of those it was, but it is definitely an ongoing, deepening response with no other therapy being given during this period. We've also been able to see in some of these patients that the NK cells have ability to migrate to tumor locations. And in this case, actually to leukemia that was in the brain. This particular patient on the top had residual leukemia around the brainstem and the lateral horns. About a week after the NK cell infusion, other step changes. You got another image, and it showed all this inflammation around the places where there had been prior disease. Got some steroids to calm that down, and the inflammation went away. He got better. He came out of the ICU, went home, and all that was left were these kind of punctate scarred lesions there in the brain. So suggesting again that the NK cells go to sites of inflammation, of danger, help set up an inflammatory response there that then can be either continued with NK cells or transferred to the adaptive immune system. And this is a young patient, a 2-year-old, had a AML chloroma. And again, you can see evidence of inflammation around the site of the cancer, and then just little bits of scarred residual disease afterwards. So to this point, we have completed a number of trials with haploidentical NK cells. We've also done 2 trials with autologous NK cells. Overall, over 200 infusions and over 100 patients, and these have been doses up to 10 to the 8th NK cells per kilo given IV, and in some cases as many as 6 doses at 10 to the 8th per kilo. So we're really able to deliver a large number of cells and sustain a cellular response to this cancer. And there are a number of ongoing trials, both in our pediatric program and the adult program, around leukemias, sarcomas, carcinomas, neural crest tumors, and brain tumors, lymphomas. I think a wide variety of applications. So the barriers to this are really multifactorial. So there's, first of all, the cost. Right now, our cost is around $35,000 each time we do one of these expansion processes in the GMP facility, and that gives us anywhere from 1 to 6 doses of therapy. There's some question of quality. There are some manufacturing failures. There's times when we have contamination. There's times when our endotoxin doesn't meet release criteria. We try to get a very low T cell content so that we're not at any risk of GVH, and that can fail. There's some time and logistics issues. If we're doing patient-specific manufacturing, we need 3 to 4 weeks to do the donor testing, the manufacturing of the cells themselves, and the release testing. And sometimes patients just can't wait for that period. And there's some logistics with the GMP facility. They need to have a slot that's available when the patient is available. And even though it's not exactly needing to be available that day, like for a fresh infusion, it still has to be within the right kind of timing so that we can get a donor in and the NK cells made in time for the patient. There are some issues with dose. If you're using heavily pretreated patients and trying to do an autologous NK cell, then some of those patients we've shown have really poor expansion, that cells just don't grow well enough. Even if you're doing it from a normal healthy donor, you may not be able to collect enough starting material for a large number of doses. And some donors are kind of unpredictable. They just happen to be donors that don't expand as well as others. And last is around potency. So trying to develop a drug that has consistent potency, particularly if you want to go to the point of commercialization and marketing and meet FDA requirements for measuring consistent potency. Not all patients or donors, not all patients have an ideal family donor. Not all donors have the same potency, and not all tumors are recognized through the classic NK cell receptors. So the predicting potency for an individual patient is very difficult, and finding the right donor that actually meets all those criteria is not entirely straightforward. So we've progressively moved over the last 10 years from autologous patient-derived manufacturing to an allogeneic universal donor approach in order to actually overcome all of those hurdles. So with an allogeneic approach, you never have a problem of not having a donor. You've already got donors that are frozen and ready. You don't have the cost issues because you can now actually leverage issues of scale and create very large batches that treat 20 to 50 patients per batch. The complexity of the matching piece is less, and of GMP resource utilization. So you can actually plug in your manufacturing whenever you have a lull in your GMP space. You don't have to do it based on patient need. You can just be manufacturing ahead of time. They're immediately available, so your patients now don't have to wait. And you now have almost no dose limitations. You can really go up as far as you think you want in both individual doses or repeated doses until you hit something that's intolerable, which is something we actually haven't done yet in our NK cell therapies. And you can select your universal donors for those that have the highest efficacy. So you've really pre-screened all of your donor characteristics in a way to maximize potency. So we've really just done the first of these. We've now treated 7 or 8 patients on a leukemia trial, and are in an adult leukemia trial. We've treated 1 patient on a pediatric leukemia trial using this universal donor approach, and we have several sarcoma studies that are just getting ready to treat their first patients. But we have a couple of patients where we've been able to track infusion of the cells. And you can see they maintain very high levels of these NK cells in the peripheral blood for the 2 weeks of where they get the infusions, and then it drops off a little bit afterwards, still detectable, but we're able to really use this, like I said, as a drug where we're managing an area under the curve as sort of drug exposure. And they're definitely persisting in this lymphopenic environment. So now this really enables us to think about CAR NK cells, which is the primary focus of this talk. If you're going to go to a different cell type than a CAR T cell, one of the things you want to solve is the very high patient-specific manufacturing costs, something that we've already seen in the NK cell field, but is made log orders worse by trying to do genetic modification on a per-patient basis. So I don't really need to introduce much about CAR design. Most of you who are listening to this will know this quite well, but you're basically trying to combine the targeting of an antibody, the variable regions of the heavy chain and light chain of an antibody, and link that to the signaling of a T cell. So normally a T cell would signal through alpha beta receptors, where it would find antigen, and its co-stimulatory molecules, including CD3, would provide your activating motifs for signaling into T cells. Well, a CAR and an NK cell is really not any different. NK cells also have CD3 zeta. They also use CD3 zeta for signaling through some of the activating receptors in an NK cell. So a CD3 zeta signal is a native signal for an NK cell, and many of the same CARs that work in a T cell have been used to work in NK cells as well. You all know that the CAR T cells have really been a huge benefit to treating leukemia and now lymphoma, and have very high levels of efficacy. And part of the move to consider NK cells is to be able to leverage the universal donor approach and get away from patient-specific manufacturing, but also to try to think carefully about situations where you don't want very long-term targeting of against the antigen for a long time, like you would get with a T cell. You wanted that to be able to be more transient and more controlled in a dosing kind of way. So the hurdle for this has been genetic modification of NK cells. And while T cells can be modified by a large number of approaches, viral approaches, transposons, plasmids, mRNA, electroporation, the same approaches that work well in T cells generally do not work in NK cells. And this was actually took us a while to figure out why. But if you think about sort of the core normal response role of NK cells, which is to be first responders against viruses, they are quite resistant to infection by a variety of different viruses because they have large levels of TLRs and PAMPs that are really built to recognize CBG motifs and double-stranded RNA and other elements that are signs of non-eukaryotic DNA inside. And so an NK cell would rather, speaking a little bit anthropomorphically, they would rather die than be infected because as a first responder to viral infection, they need to be resistant to those viruses so they can last long enough to set up that inflammatory response that's needed. So traditional approaches that have worked for T cells have generally resulted in very low transduction efficiency and a lot of cell death. And we're really left with just transfection approaches such as approach with electroporation of plasmids or RNA, which are all very transient and need to be sort of makes it difficult to commercialize on upscale. So we worked with a group in Canada and compared quite a few different lentiviruses and found that one of the reasons that the lentiviruses were not working as well as they did in T cells was expression of the receptors for the lentivirus. So the lentivirus ought to be able to bypass some of those mechanisms of viral recognition, but in particular, the VSVG that is most commonly used for CAR T cells has very low transduction efficiency in NK cells, and that's because the receptors that it primarily uses are quite low in terms of expression. There's a couple that are expressed a little bit in the LDLR family, but some of the most utilized in VSVG are low. Now, RD114 is a little bit better. It uses a receptor that is upregulated by activation, and so you can get a little bit better transduction. But like I said, it's usually 20-ish%, maybe less. And we could get better with a baboon pseudotype lentivirus that had been described about 8 years ago because it recognizes both types of ASCT1 and 2. So the ASCT receptor, it has two isoforms. RD114 only recognizes one, but BAAV recognizes both. Oh, sorry, my slides are kind of going on their own. So anyway, there are some approaches that may be able to make this better. BAAV's been a little bit difficult to work with and so far has not been put into clinical trials, but there are several groups that are actively working on it and is being sponsored by a couple of companies that have been working with BAAV to get this into the clinic. And so transduction rates with lentivirus might be improving in the future for NK cells, but at the moment still rather difficult if you have to use VSVG or RD114. So nonetheless, those have been used for creating CAR NK cells. And in the paper by Katie Rezvani and her group at MD Anderson, they used a retrovirus with the RD114 pseudotype to transduce their cord blood-derived NK cells. And they were able to see some important responses, both MRD positive and MRD negative responses with an overall rate above 60%. The persistence of these NK cells with the CAR transgene was measured, but it was reported in terms of copies per microgram of DNA. Now, if you go back and sort of recalculate this in terms of a cell percentage. This is in the blood, and a human cell has about 6 picograms of genomic DNA, which by calculation means that at the 10 copies per microgram, you're at a 6/1000th of a percent presence in the peripheral blood. So you can see that this is at their peak. The very peak level that was detected in these patients is still only in about the 1 to 5% range for numbers of NK cells that are there in the blood. Now that's still not terrible, but this is the peak, and it was the peak in the first 14 days, but then trailed off afterwards. And be able to transduce larger of cells, and third, to get those cells to continue to last longer for better responses. So I worked for a while actually on NK cell transduction, had several collaborators, as well as working under Lawrence Cooper about 10 years ago, where we were working on transposon transduction of T cells, and we really couldn't get consistent NK cell transduction of a CAR with transposons, with lentiviruses, with retroviruses, or stable transduction with others like AAV, which is not going to be integrating. And so I kind of gave up for a while any of the NK cell CARs, but turned instead, hired somebody here, Mason Kararuti, who had some experience with CRISPR, and started to look instead, could we knock things out? And so CRISPR, as you know, has really changed our field in science, received Breakthrough of the Year designation in 2015. And its primary role is to identify a gene that you want to delete and induce a double-stranded break in that gene. And then by mismatch repair and errors in repair, then you actually delete the function of that gene. So we worked a little bit at how to do this in NK cells. And of course, we weren't going to be able to do this by traditional viral transduction methods, by the same reason that I just explained we couldn't get gene transduced in. And he solved this by instead creating complexes of Cas9 and RMP. And right after you activate the NK cells in the proliferation and expansion process that we developed, you turn on a bunch of the DNA repair genes that allow you then to get the Cas9 RMP in and initiate that repair event. So a combination actually of the fact that we had learned how to grow the NK cells, as well as this new approach for pre-complexing guide RNA and protein, really enabled this approach. So the protein itself doesn't trigger any of those TLRs or PAMPs in the NK cells. It's just protein. And the guide RNA looks more like human RNA. It's not double RNA. It's not complexed in a way that makes it easily recognized, and it only needs to be transient. So it will be in long enough to do its editing. And so with this, we could pretty consistently get 80 to 90% editing efficiency. So we worked with a group at Johns Hopkins to look at a sort of proof of concept approach, which was to solve a problem that had been with antibody therapy, particularly with daratumumab to CD38. Now, CD38 can be used to target multiple myeloma. It is an approved therapy for that disease, and it does so by binding with the FAB end to CD38 on the multiple myeloma surface, and then an NK cell sees the opposite end with CD16, which then triggers an activating event, release of granzyme and killing of the multiple myeloma. The problem was fratricide, that it turns out that CD38 is also expressed on NK cells themselves, and so the NK cells kill each other through the same antibody that they're trying to use to kill the cancer. And so we hypothesized that if we could get rid of CD38, we could solve this, and that the NK cells would actually then go on to continue to be able to kill the multiple myeloma itself. So first of all, the knockout, as I mentioned, quite efficient. We could easily get 90% of the NK cells to be knocked out. We did some additional purification to try to sort them. They expand just fine. So knocking out CD38 didn't change our ability to grow large numbers of them. And you can see very clearly that when we adoptively transferred them into the mouse and added daratumumab, if you transduced CD38 wild type NK cells, then the moment you gave daratumumab, they all killed each other and you lost all your NK cells. Where if you knocked out CD38, the NK cells would stay and they would no longer be eliminated by fratricide. This helps then in ADCC. So if your cells can last longer, you have better killing across a wide variety of cell lines and also in murine models. So what this now allowed us is an opportunity to think about knocking things back in. So where I had kind of given up on editing NK cells and putting in a CAR, we now had an opportunity to do site-directed knock-in. So with CRISPR by itself, you're really just trying to knock things out, and you get this disrupted DNA. If you can do CRISPR first and then add in a template where donor DNA matches the DNA sequences of where you just edited, you can now insert or repair DNA here. And this actually was made possible by the fact that we had a group by us that was working on AAV. And as many of you might know, several of the new gene therapies using AAV for spinal muscular diseases, including SMA, came out of Nationwide in their development. And so there was a very nice core group of people to help us designing the AAV vector to get that in. And so we ended up with, in terms of an approach, was first using the Cas9 RNP to target a location where we wanted to insert the CAR transgene, delete that piece, and then very shortly afterwards, transfect those NK cells with AAV that had the CAR vector, which would paste into its site. And now instead of transducing that target gene, you'd be transducing the CAR. As I mentioned, we used AAV vectors from collaborators here at Nationwide that had developed this for spinal muscular atrophy. And now we've actually shown very nicely, so this paper just came out a few months ago, that we can get very
Dr. Lee, I think we suddenly lost you. Can you?
Dr. Lee, are you there?
Now, although NK cells can target AML quite nicely by themselves, there are definitely some cell lines and patient samples that are resistant. In this case, a cell line above this is HL60 as a target cell line. The one below here, this is a patient-derived leukemia sample. You can see the NK cells by themselves really have modest ability to kill the leukemia cell line, but the moment you add the CAR, you have much better directed recognition through CD33, which is on many AML samples. We've done some additional typing of this to show that the CAR NK cells actually target a specific population that is left over after NK cells alone. So they're actually not just enhancing overall killing, they're redirecting the NK cells to targets that would otherwise have been missed by a native bare NK cell. And we've done a variety of other NK cells with CARs to CD38, to GARP, to CD70, HER2. In the last year, we probably made a dozen different CAR NK cells using this approach. So the last part of this talk, I'll just finish up here talking about where you put the CAR. And although we now have some very nice methods of putting the CAR in, I think we need to be careful about thinking that the NK cell itself is just a big bag of perforin and granzyme that we're conjugating to an antibody. And in many cases, it feels like the field has gotten so excited about the fact that NK cells have all this perforin and granzyme that it doesn't really matter what NK cell we use. It's as long as it has perforin and granzyme to release a payload. And that really is thinking like this antibody toxin conjugate, like whether you put ricin domain or a calicheamycin onto the antibody, or whether you put a bag of perforin, it doesn't matter. It's really just an antibody with a death domain. And I think there's much more to it than that. We really have to understand NK cells and NK cell CARs. Yes, there's a lot for us to design in the CAR structure around targeting and signals and the editing approach, but the cell base really does matter. Where did this NK cell come from? Did it come from an iPSC, a cord blood, a peripheral blood? Because all of those have different homing molecules. They have different inhibitory domains that are present. They've been cultured in different ways that imply something different around their level of senescence. What cytokines were used for expansion is going to matter, whether it's IL-15 or IL-21, whether we include TGF Beta. I'll get a little bit to that in the end of this. Or nicotinamide. There's a whole group around enhancing these with nicotinamide to make a better NK cell base. Whether we do this as a patient-specific NK cell or an off-the-shelf or a patient donor-derived, all of those are going to impact this piece of the drug, right? If you think of CAR NK cell as the drug, and those have huge impacts in everything that you want to do with the cell about homing and targeting and persistence and overcoming the microenvironment. So one of those major pieces, particularly for solid tumors, which is a lot of the emphasis that NK cells have received in the last couple of years, is around TGF Beta. And TGF Beta is a really dominant signal in most solid tumor microenvironments when it comes to understanding cell therapies and approaches to using T cells or NK cells in a targeted way. So particularly for the NK cell, TGF Beta has a large number of things that it does. It downregulates some of the activating receptors such as NKG2D, NKP30, and DNAM. So you have less that the NK cell itself can trigger itself with. It decreases expression of TRAIL, which was a major effector molecule that mediates cell death of the target. It decreases CD16, which means that it's less able to interact with antibodies, and it decreases cytokine production so that whole process of interacting with adaptive immunity is now hindered. So TGF Beta is really important for us to try to overcome. It's in a large number of cancer types. This is looking at overall TGF Beta expression across over 1,000 different cell lines in different categories. And about the only ones that don't express really high levels of TGF Beta are small cell lung cancer and prostate cancer. The rest, typical tissue expression, this is in a log2 scale, is down, problem here, is down around a 1 to 2. So even by the time you get to 4 or 5, you're already 10 times higher than normal tissue. You're at 100 to 500 times higher than normal tissue in these very strongly expressing tumor types. So TGF Beta signals through two different pathways. Signals through a canonical pathway, which is through SMAD3 and SMAD2, mostly, and through a non-canonical pathway, which we know a lot less about. We know some of the genes are involved. The SMAD3 pathway is a little bit better to understand in terms of knowing that it's the complex of phosphorylated SMADs that then become a transcription factor and activate TGF Beta target genes. So we found out somewhat by accident that if you imprint cells with TGF Beta or include TGF Beta in the culture media while they're growing, they actually completely downregulate SMAD3. And so they lose that entire canonical signaling pathway. And this happens at both the protein and the RNA level. And what this results in is an NK cell that is no longer sensitive to TGF Beta and actually has higher levels of cytokine production rather than lower levels of cytokine production. This process results in some epigenetic remodeling, and there are really hundreds of genes that are differentially affected by this, and particularly genes that are related to holding on to these secretory granules that have perforin and granzyme and cytokines in them, so that they're actually much more fit for repeated killing in a serial killing kind of manner. genes that are related to oxidative phosphorylation, so in terms of maintaining metabolic fitness, and addressins, and this is really quite important in terms of migration. So the addressins that are expressed on these cells after the TGF beta imprinting are much more like those that enable tissue homing than those that stay in the blood. So as I mentioned, what base you put a CAR NK cell on really matters depending on where you want it to go and whether you want it to stay in the blood, go to lymph nodes, go to solid tissue, particularly tumor tissue. We may need to look at a variety of different kinds of cell substrates that we're genetically modifying. We're doing some work in testing this in canines. We have a clinical trial where we've been able to develop the same process for making TGF beta imprinted NK cells in humans. Works for the dogs. And so we're testing this in some sarcoma models, osteosarcoma in particular, and also looking at building onto this in our CAR NK cell approach so that we can generate canine CAR NK cells as a large animal model before we get to FDA approval. Our first dog was infused actually a couple of years ago. We're now up on the 3rd dose level for this. It's really been tolerated quite well and some encouraging early results. So last thing I'll say, just around how do we get them to last longer? We were really hopeful that lymphoid malignancies that was out of MD Anderson included IL-15 in the transgene. And although there weren't really high levels of NK cells shortly after infusion, they were able to find them quite a while afterwards. High levels of IL-15, though, there's some recent studies by the group at Hopkins that has shown quite nicely that the IL-15 itself can be quite toxic. And so we're going to have to be pretty careful about what our constructs look like and how these cytokines are regulated in order to get a maximal benefit for the CAR NK cell without overdoing that and getting unexpected toxicity to the patients. So with that, I'll finish and take some questions. I think we're finally at a place now where development of NK cell expansion and gene modification approaches, we need both. We have to be able to grow the NK cells and we have to gene modify them. We now have both of those sufficient to really think of CAR NK cells as a viable commercial therapeutic. And future development of this is really going to be critically dependent on tumor homing. The NK cells won't do anything if they can't get to the tumor. Tumor antigen targeting, which is the whole promise of the NK cell CAR to begin with, that the CAR will enable that targeting. Overcoming the microenvironment, a variety of approaches, including what I just described around TGF beta imprinting, and how we integrate this with existing therapies, because it's not likely this is going to be a standalone cure on its own. It's going to have to integrate with, at the very least, a conditioning chemotherapy, the way that CAR T cells are given, maybe not just lymphodepletion, but pre-treating the tumor itself or the tumor microenvironment. So I put together a list of some papers here. I'll help distribute this list through the sponsors. I selected just a couple of papers that were either all within the last year, within more like the last 9 months, and specifically those that are by some of the groups that I think are kind of leading this. There's a group in Spain that has really been quite active in developing CAR NK cells over the last 5 to 6 years. Stephanie Wen, this is a group in, I believe in the UK. Chalice Bonifant at Johns Hopkins University. Michael O'Dwyer, who is also in the UK and leads a spin-off company there. Rizwan Romi, who is at Harvard and came out of the group with Washington University. And then Katie Rezvani that we've mentioned at MD Anderson. Some early studies, as well as most of these are kind of overall reviews to give you a flavor for where we think CAR NK cells are going. And then lastly, so 6 years ago, Nature Biotech in its front page said, news, NK cells are going to be the next big thing in immuno-oncology. And maybe now 6 years later, it's CAR NK cells will be the next big thing in immuno-oncology. We'll have to see where that goes. With that.
Yes, thank you, Dr. Lee. Thank you, Dr. Lee, for that very detailed and exhaustive presentation about NK cells and CAR NK cells. The floor is now open to questions. So if you have any questions for Dr. Lee, please, now is the time to submit your questions to the Q&A portal. Dr. Lee, I'll start the question and answer session with a more general question. And this comes from my lack of in-depth knowledge in the field. So my understanding is that T cell receptors recognize cancer cells, and that's, triggers the cytotoxic T cell mediated cytotoxicity. How do NK cells recognize cancer cells?
NK cells, rather than recognizing specific cancer-related antigens, the way the T cells do, they recognize antigens that are normal proteins that are upregulated in response to stress or danger. So they're more about looking for overall signs of danger than they are about specific proteins. And those are members of the MICA and ULBP family, the poliovirus receptor family, and there's several others that are often upregulated in virus-infected and cancerous cells as a response to stress, whether that's metabolic stress or genotoxic stress, or the stress of chemotherapy or DNA repair because of radiation. All of those events tend to upregulate the danger signs that NK cells recognize.
So that goes in with what you told about, NK cells being the first responders to virus, and also NK cells responding to both virus and cancer. I was actually asking about more specific, are there any receptors on the NK cells that recognize these? Like you have the T cell receptor, which is like this classic receptor.
Yeah. So the receptor that recognizes the MICA and ULBP family is NKG2D. The receptor that recognizes PVR and its relatives is DNAM. There are receptors for HLA that are in the KIR family, a family of about 15 receptors activating or inhibitory. CD16 is the receptor that recognizes antibodies. If you have an antibody stuck on a cell, that should be a danger sign. You shouldn't normally have antibodies stuck to your cells. So that's through CD16.
Yes, so
30 or so receptors altogether that NK cells use to make that decision.
So this then goes with the concept that NK cells have activating receptors and also inhibitory receptors, and there's a balance between the activating receptors and the inhibitory receptors that decides whether NK cell is going to perform cytotoxicity via perforin granzyme or it's not going to. That's pretty much where you're coming to. So actually that brings me to the next question that I have, and this may be jumping all the way to the end of your presentation. You did mention that you have TGF beta, which you found, and having TGF beta in the culture medium somehow seems to get rid of these balances between the activating and inhibiting and overcomes the signaling and helps the NK cell to perform a cytotoxic effect. Am I understanding that right?
It stopped part of the signaling specifically through the TGF beta receptor. So there's a large number of inhibitory receptors. This process really only influences that one of TGF beta. There's plenty of others that we need to look at and find ways around. TIGIT is an inhibitory receptor that might be worth knocking out. Several groups have done that. PD-1, maybe not. It's certainly it plays a big role in T cell therapies, but it's not as clear that it's important for NK cells. So I think each of the negative receptors are going to have to be addressed separately.
Okay, so that comes actually to the question that I wanted to have. It looks like the biology is actually a little bit complicated. So when you're programming NK cells or even CAR NK cells, how do you account for these differing signals to make sure that you're activating the NK cell?
Yeah. The it's not so hard in terms of CAR design because you just focus on putting the activating
Yeah.
signaling motifs in the CAR.
Yeah.
When you're building the NK cell, like again, this is back to that idea of the base that you use as your NK cell therapy, right? Where it comes from, how did you grow it? Each of those different expansion approaches and sources may have different relative contributions of those inhibitory pathways. So it's one of the things for our universal donor that we really focus on individuals who have mass licensing, so that the activating piece of an NK cell is most turned on. But there's still a lot we need to know about those. We may find some ways that we can culture NK cells that it won't upregulate, say, TIGIT and PD-1. We may just have to knock them out in order to overcome them. But all of those will create a new base on which to build a CAR NK cell.
And could you tell a little bit about what you are particularly looking at to, you did mention TGF beta, but is there anything else that you're looking at to activate or regulate the activity of expansion and activity of NK cells?
Yeah. IL-21 is being really important for expansion. We're learning a lot more about it, that it's actually quite important for metabolic fitness and for downregulation of some of the pro-apoptotic pathways. I mentioned the TGF beta piece. There's a whole lot of biology there that we're trying to understand. I do have a graduate student who's working on TIGIT and trying to identify ways to overcome that. There are some potential ways to design the NK cell CAR itself to be more NK cell friendly. Most people have used T cell CARs in an NK cell and they work, but there's a lot of signaling endodomains that might actually be better. Can we get the signaling domain to not be inhibited by CHIP1, which is what the PD-1 and inhibitory cure pathways use? Could we make the signaling domain also have a cytokine signal? Could we have it additionally signal co-stimulatory molecules that are more important? We think 4-1BB is probably more important for an NK cell than CD28. So there are some of those design pieces that would be appropriate, I think, to try to overcome some of those pathways.
So you're talking about the second, third generation, and fourth generation CARs more specifically, right? I mean, so it really
yes, and
looks
developing
like
those for the NK cells rather than just using what looks like it's good in a T cell.
So it really looks like there are many different pieces that you have to consider while developing an NK cell. Is it reasonable then to expect a universal NK cell, like model, or do you see it more as like understanding the end use of it and then culturing the NK cell tailored for that specific use? And the reason I'm asking this is because we hear a lot about the shelf life of NK cells being really good. But my naive understanding is that there are so many pieces that you have to consider. What is the point of having a generic NK cell when it looks like a more tailored approach seems to be more efficient? Maybe I'm wrong, but I'll let you address that.
Yeah, no, I think if we really, really understood everything we needed to know about the biology of a particular person's cancer, we might be able to find a fit for purpose application that custom designed the perfect NK cell for them. I think we're a long ways from that. And so where the field is at the moment is trying to understand the NK cells and then figure out where they fit in patients. So there may be relative advantages and disadvantages of IL-21 expanded cells versus NAM expanded cells versus cord blood versus iPSC. And for the time being, those are the basic approaches that are going to provide a universal donor approach, and we're going to have to see which diseases they fit best in. When we know more, we can go the other direction. But I don't think we know enough yet to say you have cancer X, therefore I'm going to pick cell Y.
Yeah, so that actually makes sense as well. And you try to understand as much about the disease biology before you can go into designing more specific things. That also brings up another question in my mind. You did mention about CAR NK cells and one of the applications of CAR NK cells in solid tumors.
Mhm.
And usually we use CAR cells for hematological cancers, and there is this whole area of getting penetration into the solid tumors and also the immunosuppression of the tumor environment. Is there anything specific about NK cells or CAR NK cells that makes them, overcome these challenges and that can be used in solid tumors?
I think there's some evolving understanding of a group of NK cells that are referred to as being tissue resident. We know that NK cells are present in lots of different tissue types, and the more we understand those, the more we know what gets them to that tissue type. Why is this particular NK cell found in the liver? Why is this kind found in the gut? And I, I think we'll find that there are ways for us to manipulate cells to obtain those characteristics, so they will go to the right kinds of organs. And if they're found in a particular type of solid tumor, whether it's a epithelial carcinoma or a deep tissue sarcoma, we'll know better about what gets to that kind of tissue, so the NK cells can go there.
Okay. So for right now, they're used in hematological cancers, and
They're used,
that's
yeah, they're used more in hematologic cancers because that's where the original evidence was that they mattered from a bone marrow transplant perspective.
Mhm.
Um, and so it was easier to apply. And hematologic cancers are in the blood, so it's easier to infuse cells into the blood and get them to work. I don't think that's it's used more in hematologic cancers because that's where it was obvious to go, not necessarily because that's the only place that they that they have application. It's just it's a harder crack to get to the solid tumors.
And then I have a more specific question related to the your slide where you talked about NK cells, and you used the expansion of NK cells using the feeder cells K562 IL-21 cell line feeder cells. And you were talking about expansion, and it I don't remember the exact number, but you were getting a lot of cells that you were getting from that particular methodology.
Uh-huh.
And how much of the actual cells do you then use, and what is the shelf life? How do you store it? Could you tell us a little bit more about what happens to
Sure.
those cells?
Sure. So in our universal donor platform at the moment, we are doing an apheresis collection of the donor, and that is being split into 20 to 50 fractions and cryopreserved. Then we take one of those fractions out and do the NK cell expansion. And our target product is to generate 50 billion cells from that one expansion. And that is then frozen down into multiple dose aliquots, so sufficient for somewhere between probably 10 and 30 doses, depending on whether it's adults or kids. When those are frozen down, as far as we know, their shelf life is basically indefinite. And so we can generate hundreds of doses from a single collection of a donor.
And so this is like an allogeneic NK cell
Uh-huh.
for allogeneic. And you did mention something about cryopreservation making them more viable. And I was wondering if you know the biology of what is happening that makes them viable after cryopreservation. Because you would assume that cryopreservation actually makes cells die when you go through freeze thaw.
Mm-hmm.
But how do they then have this increased viability?
Yeah, so I don't know that we really understand very well why freshly isolated NK cells don't cryopreserve well. T cells cryopreserve fine, and at least microscopically, you look at them, there's not really much difference between the two. There's a couple of possible hypotheses that probably still need to be tested. One is large amounts of granzyme. When you cryopreserve, you damage membranes, and perhaps the granzyme is being released intracellularly and killing the cell. But that doesn't explain why it doesn't happen in T cells. There's a large release of some proteins that are in the metalloproteinase family that get activated by cryopreservation. That could lead to cell death. I think we don't really know yet why it is that if you expand them first, they actually freeze fine. But if you try to freeze them when they're fresh, they don't.
Okay, so but I think it works. So you go with it, and sometimes in science you do discover things serendipitously.
Yeah.
I have a more broader question for you now. So where are you in your research? Are you looking at clinical trials? What's next?
We have a bunch of the trials that are just now starting with the TGF Beta imprinted NK cells, and so that we'll be learning a lot about those in the next couple of years. And our first CAR NK cell trial is we're finishing up kind of process development and manufacturing for that in the GMP and protocol development. We hope to have that open early next year.
Do you have any papers that our audience can find to see the progress of the clinical trials?
Yeah, there's a couple that we've done recently. Most of them were cited in the slides, but 2 papers by Stefan Churia for the transplant trial. Lucia Sela published her paper in British Journal of Hematology last year. One paper on the brain tumor study was by Suman Katua in neuro-oncology, and there are 4 papers that are in progress that we haven't published yet. The neuroblastoma trial, one of the other leukemia trials with Stefan Churia, and a couple others that should come out in the next 4 to 6 months.
Thank you, Dr. Leeds. Thank you so much for such an illuminating conversation and for answering the questions. It is truly fascinating, and immunology is fascinating, NK cells are by far so more fascinating. I do hope that we get to see more of your research, and wish you all the very best. Looking forward to see more of your research, actually.
Great.
Unfortunately, that's all the time we have for today. If you have any further questions, please consider reaching out to Dr. Lee directly. His email is shown on the screen. As a reminder, the webinar will be archived on the DDN website, and you will receive an email notifying you when the webinar is available on demand. On behalf of DDN, I'd like to thank our speaker, Dean A. Lee, as well as the sponsor, BPS Biosciences. And of course, a thanks to you, our audience, for listening. Goodbye. Have a wonderful rest of your day.
Thank you much.